It all looks the same until you take a closer look. Today, we're comparing 3 of America's most popular ketchups, Heinz, Hunt's, and Great Value. At first glance, they all seem identical. So today, we're putting each ketchup under the microscope to see what's really hiding inside. We'll introduce each one to live rotifers and watch how these tiny freshwater animals respond. By the time this video is over, you'll see which ketchup looks the cleanest under the microscope, which one leaves behind the most visible plant material, and whether paying more actually gets you a different product. Let's do this. Here is a sample A rotifer. I'm not going to tell you which ketchup this is until the end, guys, so try to guess in the comments what you think the ketchup is. So this is our rotifer, and a little bit about this guy before I introduce the solution, but you see those tiny hairs in front of his mouth? Those are called cilia. And what that does, it goes in different opposite directions, and it creates a vortex where it sucks everything in like a vacuum cleaner, where he eats algae, microscopic debris, and then it goes into the mastix, which is right behind the cilia, and that's his jaw, and it grinds everything up, and it goes down to that thing moving right there is his muscle. Basically, it pushes the food into his stomach, and basically how he holds on is his toe. And what he does is he grabs onto a piece of debris, he stretches out, and he's doing what he's doing. That's pretty cool, right? So I'm gonna go ahead and introduce the solution. So watch the bottom right side of your screen or TV, and you'll see the sample A ketchup I mixed together with distilled water and introduced it on the right side of the cover slip. So here it comes. Now you'll see a jolt from this guy, and then from there, you'll see a big rush of this stuff coming in. So watch him. So boom, right there. Okay, I just introduced the solution. Now here comes the tsunami. Okay, boom, there it is, you guys. That's a wave of 3,000 gallons of ketchup mixed with distilled water. So watch his reaction. First line of defense for rotifers is when they curl up into a ball, they protect themselves like a turtle, and they'll stay there until the environment is improved. But he looks pretty good. Yeah, nothing's really changing for him, and he's still foraging, still eating. That's a good sign. I'm not really expecting anything crazy, but this is just a visual to see the stuff that we eat every day and how microorganisms react to it. So let's go ahead and move over to sample B and see how that rotifer fares. Okay, it looks like I found 2 for sample B, and it's pretty cool how they operate. You notice his cilia is moving and sucking in all his food nutrients. You'll see his muscle moving there. So let me introduce the solution. Sample B ketchup mixed with distilled water. Let's watch his reaction. Right side, there it is. You see it, guys? Just got blasted again. 3,000 gallons of the sample B ketchup with distilled water, and so far so good. I don't see both of these guys going into a ball. He's still eating, as you can see. I mean, he noticed his environment's change, and you see that? He's changing locations. He's not sure what's going on. See, he's testing the water again, and let's see how it reacts and what happens. So I fast forward a good 10 minutes, and both of them are doing very well. So no curling up in balls. They're not swimming everywhere, going crazy. So sample B did pretty good. Let's go ahead and move to sample C rotifer and see how he or she does. So here's sample C rotifers. This is pretty cool. 40 times magnification and 1 drop of water from my flower pot outside. And look at all the debris in here. Look at all the life. It's incredible. In this slide alone, I think I have roughly 8 or 9 rotifers that I found. So I just wanted to show you guys what contains in 1 drop of water in your backyard in a flower pot. So before I introduce the solution, I found a little guy darting around everywhere. You see that guy up there on the top of your screen or TV? That's called a paramecium, and he's a little guy, but very, very fast. He's got cilia, the hairs that you see on the rotifer's mouth. It's all over his body. And what he does is it acts as paddles, like 1,000 paddles in the water, and he darts in and out, and he eats bacteria. That's his primary food source, tiny bits of decaying plant material. He eats microscopic algae, yeast cells, and other very small protozoa, which is the same species. So I'm not sure if he's a cannibal or not, but I got very lucky recording him because usually they're so fast. Even at 40 times magnification, you can't keep up with them. So this is pretty cool. So back to the rotifers and the blast of the ketchup. Look on your right side of the screen, you guys. You see that? I just blasted him all the way across the cover slip. A little too quick, but this guy in the middle is doing pretty good so far. So where did the other guys go? But maybe we'll look for him. But he's still eating. That's a good sign. Even after a few thousand gallons of ketchup, got dumped on him, but let's go look at the other guys. Okay, where's he at? There's one up there. So, all right, he's doing okay. Looks like he's still, there's another one there. So they have not went into a ball, not as of yet. So obviously their environment changed and they're trying to adjust, and he's still eating. That's good. And I don't know where the protozoa went. This guy's so quick. I'm not sure. I think he got blasted off too, but here comes some more right there. But so I found one over here on the other side of the slide, and he or she is in a ball. So that's their first line of protection. I'm not sure what's going on with him. Obviously, thousands of gallons of ketchup, obviously, but this is their first line of defense. So the other samples did not do this, but this one is. And I do know the amounts that I introduced were fairly the same. Some were quicker than the other, but okay, so let's go ahead and end the Rotifer test, and let's move over to the microscope, and I'll show you guys what ketchup looks like under the microscope. Let's go. So here we are at 40 times sample A. Guess which one this is. And right there, I'll come back to that, but that's the plant material. I think it's tomato skin, but I'll get a close-up on that. We'll take a closer look. But I'm looking for like debris, plastics and fibers and different things, but so far this is looking pretty good, like it's supposed to for ketchup. Again, right there, that is plants or plant. And I'll zoom in on this guy. So 100 times magnification, the tomato tissue, that's what this is. It becomes even more defined at this magnification. You can see the honeycomb pattern is made up of individual plant cells. They're each surrounded by a sturdy cell wall. It's a reminder that even after the tomatoes are crushed, cooked, and bottled, parts of their original cellular structure are still preserved. That's pretty cool. So moving on, what we're looking for, basically good signs are uniform consistency, which is the sample looks evenly mixed without large clumps. And we're looking for intact tomato cells. Seeing recognizable tomato tissue is actually a good sign. Looking for even distribution of plant material. Instead of huge chunks in one area and nothing in another. So it's good to have them spread apart. And minimal large debris, like large stems, thick skin fragments, or oversized seed pieces may indicate coarser processing. And finally, looking for stable emulsion, which is a liquid shouldn't separate into obvious, like big pools. So check out this at 400 times, and this is the plant material, which is tomato, the tomato skin. It looks like a giraffe. That's pretty cool. So let me zoom into 1,000 times, and it gets better. So here's 1,000 times giraffe skin. No, I'm just joking. This is, of course, 1,000 times tomato skin. And did you guys ever think you look at tomato skin at 1,000 times magnification? So here you go. Anyways, let's go ahead and exit sample A. Let's move to sample B and guess which one this is. All right, here's sample B at 40 times. And remember what we're looking for. Nothing too clumped together as far as the plant material. We're not looking for a lot of oil spots, So this is looking okay. Not too bad. So there's more plant material there, which is their tomato skin. And let's bump it up to 100 times. Here we are at 100 times. And again, there's the skin. So do you guys notice any difference between sample A and sample B? So take note of that. Leave in the comments what you guys think and try to guess which one this is. But again, it's looking okay. It's not, I don't see too many oil spots, and the plant material looks pretty good. It's not clumped together. So let's go to 1,000 times. Here we are. So this looks like a brain. It's actually pretty cool. But again, this is plant material. It's tomato skin at 1,000 times. So let me focus a little bit there for you. But yeah, so what do you guys think? This is pretty cool. It looks pretty uniform, not too bad. But so there you go. Let's go ahead and move over to sample C, our last sample. Cast your votes now, and let's take off to sample C. Here we are 40 times, sample C. Take your votes now, or cast your votes. So we're looking for density. We're looking for, things aren't clumped together. So this is looking pretty good. Looking for sporadic oil spots. Okay, let's move to 100 times. Okay, here we are 100 times. And what you guys think? I mean, it almost looks the same, right, to A and B. But again, just look for sparsity and some oil spots, which I still don't see. So let's bump this thing up to 1,000 times. Okay, here we are. I see a lot of debris in this one. Did you guys notice that? Oil spots, not so much. I don't see too much plant material. So take note of this. And there's an air bubble there. So not too bad, but I see a lot of debris in this field. So let's go ahead and exit this microscopic world, and I'll take you back to reality, and I'll let you know which one's A, B, and C. So let's go. We made it. So what did you guys think? I mean, they all look pretty much the same, a little bit here and there, but what'd you think of the Rotifer test? Sample A was Heinz. Can you believe it? So which one's your favorite? Heinz seemed like it was okay. The Rotifers fared pretty well with that one. Sample B was Heinz, and Rotifers did okay with that. Kind of got blasted a lot with that, but they're all right. And sample C it was great value organic. The Rotifer did not do too well with this one. So I'm not saying it's good, I'm not saying it's bad, but yeah, it kind of makes you wonder though, right? About the first two samples, you know, nothing happened to them, but in sample C, which was great value, kind of curled up in a ball and went to sleep. Oh, check out that video right here. If you want to know how Rotifers react with beer, yeah, check that one out, it's pretty cool. Don't forget to subscribe, be good to yourself and others.